Sorry, I didn't mean anything
Yeah,
personal.
okay, fine. All right, we're going to move right on to our next presentation. So,
There's actually a timer up here, right? So now we're going to use the timer.
the next company to present is Sangamo Biosciences, and presenting for them is Ed Lanphier, their president and CEO. They're located in Richmond, California. And as soon as we get the timer set, we'll start.
I'll give you 2 minutes. I got to get this.
No problem.
There you go.
Great. Maury, thanks very much. I appreciate being here. I'm going to talk about our core technology and specifically about the applications in stem cells. And so there's lots of things I'm not going to cover, but just as a very high level point, we're located in the San Francisco Bay Area. Those of you that follow baseball know the Giants won last night, so we'll take a moment to congratulate the Giants. We're a public company, been public for a little over 10 years and about 100 employees. So this is an outline of what I'm going to discuss with you. There's a lot of data in here. I'm going to move fairly quickly through it. If anybody would like any of the publications I reference, we're happy to get those to you. So what I'm going to talk about first is our platform technology. Everything that I'm going to talk about after that is really derivative of this, of how we're going to apply it. First in the area of stem cells, then specifically in the area where we're in phase 2 clinical trials in T cells, but then the application of our HIV work in stem cells, and then lastly in vivo applications. So first, this is a wordy slide, very busy, but it summarizes the work that we're doing. So we're involved in a type of DNA binding protein called zinc finger DNA binding proteins. And these are small, only about 30 amino acid modular proteins that we can engineer actually in the same way that nature engineered these proteins to bind to specific DNA sequences. And one of the things you'll see as I go through this is we can build these so that they are singularly specific to any single site in the genome, so no off-target effects. And then we can use this to bring into the context of a target gene what we would call a functional domain, a protein that allows us to drive biologies at the DNA level. So this toolbox looks something like this: the ability to target at the DNA level with an engineered zinc finger, and then either turn on or turn off the expression of a gene using a transcriptional repression or activation domain, or, and I'll spend most of my time talking about this, the ability to bring into this site nucleases that can cut the DNA. So first, I'll go back. First, we can use this to turn off DNA or turn on the expression of target genes. And then we can use this to physically modify an endogenous gene. And we do this by bringing actually 2 zinc fingers linked to nucleases, enzymes that cut DNA, make a double-stranded break at a determined site, and then we can resolve that break by either causing a break in the DNA to be resolved by non-homologous end joining, or we can use a donor sequence that we can use to correct or add DNA to that site. And much of what I'll be talking about, and particularly in the monogenic disease spaces here, I'll focus on this application of knocking out a gene in our CCR5 program in HIV. So that's the toolbox that we're employing here. The goal, however, is really much more grandiose. It's to use the exquisite specificity of this technology to go in and drive in a permanent way biology at the DNA level. And what that really means is, if you think about correcting or modifying a gene, it means that we're in a position to engineer genetic cures. And this goes, if you were here for the panel this morning, to what Dean Tozer was talking about and others, is really going beyond novel treatments to getting to the point where we're looking at curative outcomes. And I'll show you some data to support that. But what's really driving this? Why do we want to think about modifying DNA versus what's been done in the past? And I don't know if you all recognize this as a young Ernest Hemingway. It is not. This is actually a boy who was born with X-linked SCID. And if you remember back into the late 1990s, Lanphier in France, they used a Maloney-based retroviral vector to go into CD34 cells to express the IL-2R gamma gene and to try and actually cure this boy. And this is one of my favorite pictures of him. He's out in a NASA-designed spacesuit watering the lawn. I mean, this is the sort of life that this kid lives, and these are the sorts of opportunities we have to treat these kids. But the problem is, using conventional retroviruses and even today's lentiviral vectors, these randomly integrate. And what you end up with is this issue of insertional mutagenesis. You don't know where it's going to go in. But if you can target with using DNA binding proteins and actually edit the exact gene, correct the mistake in the gene, leaving the gene under the control of its own promoter, you don't have any of these issues of insertional mutagenesis. And that's precisely what we published back in 2005 in Nature. Highly efficient endogenous human gene correction using zinc finger nucleases. And as they said on the cover, genome editing, rewriting the rules of gene therapy, because no longer do you need constitutive expression, no longer do you need targeted or a random integration by a viral vector to get permanent expression of a corrected sequence. You can do this in a transient expression with very high efficiencies. So that's what I'm going to talk to you about, and that's a bit of a long-winded lead-in. But so what's our strategy? I'm going to talk primarily about the applications in stem cells, and at the end I'll try and come back and talk to you about some of the programs that we have in vivo applications. But first, starting in the topic of this meeting, the application of the platform in stem cells. We've done a lot of work across multiple targets. I'll mention some of them here. The most advanced for us is in the area of sickle cell and beta thalassemia. You all know that this is one of the largest of the monogenic diseases in terms of populations of patients, both African Americans in this country and then people of Mediterranean descent in terms of beta thalassemia. The real issue here isn't the target. There's no ambiguity about beta globin and the issues you have to drive there. The real issue is, can you correct that without insertional mutagenesis with high enough efficiencies so that those engrafted CD34 cells then have the ability to repopulate the erythrocytes in a way that can be effective. And I think that's the key thing for us here. Whoops. So really unprecedented levels of non-viral gene transfer that gives genome editing, as you can see, and this is actually the beta globin target here, at greater than 70%. And that is more than sufficient to give one the kinds of engraftment or the modification needs that one needs for engraftment efficiencies. And this is something that we've done on lots of targets in CD34 cells and shown, and I'll show you this when we talk about the HIV work, that the singular specificity of these also allow for the normal differentiation of these cells in vivo. I'll also come back to this idea of a safe harbor target as well, which is very interesting because it provides a generic site, a single site in which one can put any gene sequence into hematopoietic progenitors. So I won't go back through all of the work, but this is really the next step for us is phase 1 clinical trials. So we've also taken this outside of CD34 hematopoietic progenitors. We published work with Rudolf Janisch in both iPSCs and in embryonic stem cells. And so genome editing and induced pluripotent stem cells. We've also then taken exactly that work into therapeutic candidates. This is work that we published with Alan Brady in the UK, looking at the alpha-1 antitrypsin deficiency. This is taking a human cell, de-differentiating it back to an iPSC, correcting the deficiency, correcting the mistake in the alpha-1 antitrypsin gene, putting that back into the mouse, directing differentiation in the liver and restoring normal circulating levels of alpha-1 antitrypsin. And again, I'll emphasize this as we go through this, but transient level, so 48 hours of expression of the nuclease gives permanent correction of the gene with no integration and no off-target effects. And lastly, in terms of iPSCs, this is work that we've done and published with Harry Malik at NIH in the area of CGD, chronic granulomatous disease, using iPSCs. And again, we're able to go into, with zinc finger nucleases, in this case, into what I referred to earlier, a safe harbor site. This is the AAVS1 site. And we've now put actually over a dozen different gene targets, therapeutic gene targets, into the AAV1 site. So if you think about this from a lot of the things we talked about this morning, a regulatory perspective, same reagents, same site, same toxicology. The only thing that changes is the donor sequence that you want to put in to an iPSC. So this can be used, be leveraged many times in monogenic diseases. So that gives you a sense of where we are in terms of applying this platform in the area of both hematopoietic progenitors, but also iPSCs and embryonic stem cells. The rubber hits the road, however, when you begin to do the translational piece. And so I'm going to show you a couple of things that we're doing in the HIV area. First, starting with the target that we're going after, which is CCR5. And again, much like the monogenic disease space, CCR5 is one of these targets where there's no ambiguity about the role of HIV in terms of infection of CD4 cells. And so, and probably the best example of that, the human example of that, is the so-called Berlin patient, a man who had both HIV and leukemia, went over to Germany, received an allogeneic cell transplant from a person who had a homozygote for this CCR5 knockout, and 4 years later, he's both viral-free and cancer-free. And so the goal for us is really to mimic that both genotype as well as that phenotype, knock out both genes in either differentiated T cells or in pluripotent stem cells, replace those so that patients have a robust immune system that cannot be infected by the virus. And so these are a couple of good-looking guys. I don't know that you'll recognize them as Ernest Hemingway either. But this is Tim Brown, the so-called Berlin patient on the left. And this is Matt Sharp. And I don't know if you can read this up here, but Matt was one of our earliest patients in the trial. And this was a wonderful review on our work published in Science just a couple of months ago. And so what's the program that Matt's been through? Well, it's this program here. We take autologous CD4 cells, we modify them with a zinc finger nuclease that's very specific to knock out the CCR5 gene. We characterize those cells, and then they're reinfused back into the patient. We've now treated over 50 patients with us and our collaborators at UPenn on this, and the highest level summary of it as follows. We've seen a highly statistically significant correlation between knocking out both genes of the CCR5 locus and reimplanting those cells, and the number of reimplanted cells in reducing, actually eliminating the detectable virus in patients where we've maximized that amount. We're in 2 phase 2 trials right now looking at patients that actually have one of their CCR5 genes naturally disrupted, so that maximizes the biallelic modification. And we're in a cytoxin engraftment enhancement trial right now. I will tell you very quickly, we just presented some data at ICAC on this. I don't have time to go into it, but in addition to the antiviral effect, not surprisingly, we're seeing a reconstitution of the immune system in these patients. And in particularly, we're interested in seeing that these are these long-term memory T cells, which are the ones that are required to give and fight ongoing or renewed infections. So that's ongoing. But we've also been very successful in taking this exact same strategy into hematopoietic progenitors. We published this work early on with the goal of establishing a functional cure. This is work that is currently funded by CIRM in a disease team grant that we were funded in 2009, 2010 with Paula Cannon and John Zaya and that group at City of Hope. This is the announcement from CIRM, and we're very pleased about that. And this is just one data slide. I'm not going to go into it, but this is taking different mice with a human immune system, those mice that did not receive stem cells that were CCR5 were knocked out, and those mice that received stem cells where we knocked out CCR5. And you can see within 12 weeks, absolutely no detectable virus in these animals, normal differentiation of stem cells, and another important area in the gut mucosa, no detection of virus even outside of the bloodstream. So that gives you a sense of what we're doing both in the CD4 and T cells, or sorry, the CD4 and the stem cell space, but the technology platform is highly leverageable into areas where we can apply this. And this gives you a sense of timing that we've guided to the market. So data out of the T cell studies next year, hopefully an IND with City of Hope in 14, and potentially pivotal trials around the CD4 work in 14 and 15. So lastly, and I'm running out of time, just want to quickly cover the way we can apply this technology in vivo. We actually recently announced 8 months ago a partnership with Shire in the area of monogenic diseases, 4 targets in the hemophilia space and 1 target in the brain or direct brain in Huntington's. And this will give you a sense of what we're doing there. So it's exactly the same platform, exactly the same ability to engineer highly specific, transiently expressed zinc finger nucleases that can target any gene. The correction of that gene, much like with alpha-1 antitrypsin or with CGD and others, remains under the control of its own promoter, or it can be targeted at a safe harbor site. And this is a way of approaching this for any monogenic disease. And we have very active programs in all of these areas. I mentioned the hemophilia space. We've published and presented this work. And just to give you a sense of the experiment, this is taking a zinc finger nuclease that targets the human factor IX gene, putting it in an AAV vector, a one-time systemic injection of that formulation leads to a permanent correction and circulating levels of human factor IX in this mouse model more than sufficient to give above what is normal replacement therapies. And again, where the rubber hits the road in the biology is actually in the clotting times. And again, you can see a highly statistically significant correlation between corrected animals and those animals that don't have the factor IX deficiency. So this is largely what led to and is pushing us forward now in terms of multiple INDs in the hemophilia space. And again, the goal here is not improve treatment. The goal here is to engineer a genetic cure in these diseases. Similarly, and last slide, or last data slide for me, is we just presented data at the Society for Neuroscience meeting on our Huntington's program. I don't have time to go into it now, but this is actually a repressor target where we've created multiple zinc fingers that will only bind to these multiple repeats, will ignore the normal gene and its singular or less large repeats. And we've seen, again, genome-wide singular specificity to this target and full repression of the disease alleles. So last slide for me, just to summarize all of this. This platform is extremely robust. It can be targeted to any gene with exquisite specificity, and that's particularly relevant in the area of monogenic diseases, where there's no ambiguity relative to the correlation between the mistake in the gene and the correction. We've successfully applied this across multiple cell types, multiple disease areas, and are pushing this forward very, very broadly. It is very important as you all out there are thinking about stem cell therapies, and particularly anything you want to modify in a stem cell for therapeutic value. What's been used are retroviruses and lentiviruses. Those, by definition, in order to get long-term expression, have to insert. They insert randomly. If you can achieve the same objective with 48 hours of expression of a nuclease that gives permanent correction and leaves the gene in its own site, regulated by its own promoter, and avoid these issues of insertional mutagenesis, it's a highly differentiated platform. And that's what I said here, transient exposure, permanent modification. So thank you very much for your attention. I appreciate it.